My name is Jonathan Friedman. I'm the Development Director here at the Parkinson's Institute, and I take great pleasure in welcoming you to the series that we've established. This is, I think, the third of the series for Parkinson's patients' outreach, education, and information. The topics for today are clinical research and research in general, what we're doing here at the Institute. Many of you are patients, and you know about our clinic. The people that are not patients, we have a great clinic. And one of the interesting things we do are lots of clinical trials. I think we have over 20 going on right now. And you'll hear about some of those trials today. We're also going to talk about our research. We have clinical research, and we have basic research, and those will be explained by the presenters. My pitch also is for fundraising. I am the development director. My job is to raise money to keep things going here. As you can imagine, these days, things are rough in fundraising. People are concerned about the future. They're holding on to their money more than they have ever done before. So I ask you for your help. If you're able to donate money for our facilities, for our clinic, and for our research, we really appreciate it. But without further ado, I'd like to introduce our first speaker, Sam Goldman. Sam is a doctor. He's in the clinical research department. He says he went to the 3 best schools in the country. I'd say it's the best 3 out of 4. He went to Michigan, Texas, and Berkeley. Colorado is the other one because I went to Colorado. That's the 4th. And without further ado, here's Dr. Sam Goldman.
I didn't actually say they were the 3 best schools in the country, but I appreciate that plug. I also didn't say why I had to keep changing schools, but that's another topic. So once again, thank you all for coming today. I hope this will be useful information and interesting. And I may not be able to stay for the Q&A session at the end, so after my talk, if anyone wants to grab me and ask me questions, that's terrific. So I'm talking about what we do in clinical research here at the Parkinson's Institute. And the clinical research department is headed by Dr. Carolyn Tanner, who many of you may know. We have a very productive research program here, one of the most productive Parkinson's disease epidemiology centers in the country, if not the world. We have many completed and ongoing studies and have received funding from many institutions, including the National Institutes of Health, the Department of Defense, and the Fox Foundation. The focus of clinical research is to study the causes of Parkinson's disease, and that's what epidemiology is. And we are specifically focused here on looking at environmental factors, but also at genetic factors and at combinations of factors, so genes and environment, and how they interact with one another. We also conduct studies looking at strategies for early detection of disease and biomarkers, which I'll talk about a little bit later, as well as effectiveness of treatments for Parkinson's. And finally, we study other neurological disorders as well, in addition to Parkinson's, including multiple system atrophy, dystonia, Huntington's, or essential tremor, and several other movement disorders. So this is going to be just a very, very quick, brief overview of what we know about genetic and environmental causes and some of the work that we're doing here currently. So first, is Parkinson's disease due to genetic causes? We looked into this question directly a number of years ago, and this is a very well-known study now. We conducted a study in a large group of twins. The reason we look at twins is because if you want to know if something is genetic, twins are a perfect natural experiment. Identical twins are genetically identical. Therefore, if a disease is primarily genetic in origin, if one twin has Parkinson's disease, then the other twin should have it a very high percentage of the time among the identical twins, whereas the other twin would be less frequently affected among the fraternal or non-identical twins. So the results of our twin study, we looked at 32,000, approximately 32,000 twins, if I can make this work, from what's called the National Academy of Sciences World War II Twins cohort. They were all born between 1917 and 1927. And what we found was that there were similar proportions of identical and fraternal twins who were affected with Parkinson's disease. This is very compelling evidence that most of Parkinson's disease is not primarily genetic. Before I get to the environmental, so many of you have probably been aware that there are some genetic forms of Parkinson's disease. There are a number of genes that have been identified that can cause Parkinson's disease in either what's called an autosomal dominant or recessive pattern, so the way that they're inherited. But all combined, we only know about genes that are responsible for maybe 5% of Parkinson's disease. So this leads us to assume that most Parkinson's disease must be environmental, and that's where we focused our research. The other reason that we focus our research on environmental causes is because it's very difficult for someone to change their genes, whereas it's much simpler for someone to change their environment. There's been an association between Parkinson's disease and farming or rural residents that's been recognized for many years. There are dozens of studies from around the world that have made this association. So the question, that's the obvious factor that many farming people are exposed to is pesticides. So we conducted one of the world's largest studies looking at pesticide exposure and Parkinson's disease risk in what's called the Agricultural Health Study. And this is a study in 50,000 pesticide applicators and 30,000 of their spouses in North Carolina and Iowa. And we collected lifetime detailed pesticide use information. I'm not putting the results up of this study, but they've been submitted for publication, and we did find some very important and significant associations with several pesticides. So this is some news that should be out there within the next couple of months. Another study that we're conducting to look at possible environmental associations with Parkinson's disease is in Alaska. The reason we're looking in Alaska is because there have been some studies in populations up in the Arctic region that found a higher than expected amount of Parkinson's disease. And one characteristic that they share in these higher regions is that the diets are largely composed of marine mammals. So they eat, the individuals, especially the Alaskan native population, eat a lot of whale and walrus. And these are species that bioconcentrate persistent organic pollutants such as PCBs. So we're currently conducting a study in Alaska. We're taking blood measurements. We're taking dietary histories. And we're trying to determine if there's an association with Parkinson's disease and PCBs or some of these other pollutants. Now, this is a study that we just recently presented at the American Academy of Neurology back in March. And you can't quite see the title, but we found, looking within our population of twins that I referred to earlier, we found an important association with specific solvents. And this is a story that was picked up on the cover of Neurology Today, which is a magazine that's distributed among the neurology community, reporting that we found this exposure to dry cleaning solvents that was linked to Parkinson's disease. And the solvent is called trichloroethylene. And I'll just briefly talk about the study. Trichloroethylene is a very simple compound, and it is the most common groundwater contaminant in the United States. These are maps of several cities. This is Dayton, this is Fresno and St. Louis, I believe, and it's a very common contaminant. Greater than 50 million pounds are released to the environment annually in the U.S. It's a common occupational exposure. It's used in degreasing, cleaning computer circuitry, dry cleaning industries, and until the 70s it was used as an anesthetic. There are also a lot of common household exposures. It was used to decaffeinate coffee until the 1970s. And it's still present in numerous household products. So we looked within twins where one brother had Parkinson's disease and one did not. And then we took lifetime histories of their occupations and we estimated who was and who was not exposed to trichloroethylene, TCE. And we found that risk was 6-fold higher in the twin who was exposed to trichloroethylene, as well as for several other solvents. So this is a very exciting finding, and we've recently submitted a grant to the NIH to try to pursue this in a little more detail. So environmental causes, obvious environmental causes of Parkinson's disease, just like obvious genetic causes of Parkinson's disease, are rare. Therefore, we and most people believe that most Parkinson's is due to a combination of genes and environment. So I'm going to present one example of that we've studied here. Head injury has been looked at in many epidemiologic studies of Parkinson's disease over the years. And in approximately 70% of these studies, head injury was found to increase the risk of Parkinson's disease. But not in all. In 30%, there was no association. There are a lot of reasons to suspect head injury might be involved in a process because it can cause chronic inflammation that can actually go on for months, if not years, and it can affect many aspects of brain function, including the blood-brain barrier that could allow toxins to get into the brain. When we talk about head injury, even mild head injuries might have severe effects. This is for those of you who are not used to looking at PET scans, this is showing the glucose utilization, so it's metabolism, the rate of metabolism in the brain. And this is in a normal individual. This is in an individual who's comatose. And this is in an individual who had a mild head injury with a brief loss of consciousness. And you can see that the mild head injury and the severe head injury look identical. So severity may not be that important. What may be most important is gene-environment interaction. Some of this slide is cut off, and it's a little bit technical for me to just jump into quickly. But what we looked at here was the combination of head injury and genetic background. In particular, we're looking at a variant in a gene called alpha-synuclein. Some of you may have heard of, and I think that some of the basic scientists will be talking about that in greater detail. But we found that in individuals, so the light green bar, and I may be slightly colorblind, so don't give me a hard time about that. But the light green bar is individuals who sustained a head injury. And the differences in these bars is the gene. So these individuals had one variant of alpha-synuclein. These had another, and these had another. The odds ratio is the risk of Parkinson's disease. You can see that in individuals with this variant who had a head injury, there was a slightly reduced risk of Parkinson's disease. So the odds were less than 1. In these individuals who had a head injury, there was a slightly increased risk of Parkinson's. But in these individuals with this variant in the gene, there was a 10-fold increased risk of Parkinson's disease. So this is a really important observation, and we hope to pursue this in greater detail. So now just quickly I'll touch on some of the new research studies that and ongoing studies that we're doing here at the Institute in our department. Some of you may have heard there's now a state law requiring the reporting of Parkinson's disease. However, most physicians in the state are completely unaware of it, and there's no funding whatsoever to establish a registry. Nonetheless, we've got some small amount of funding from the Department of Defense, and we are doing a pilot project trying to identify all cases of Parkinson's disease in 3 counties in California, and this is moving along very well. And the goal of this is that later on we can try to link the rates of disease in various parts of the state with, for example, pesticide use records, which is, this could be a really wonderful resource for us. Another project is a joint project that we're doing with 23andMe, the genetics testing company. This is funded by the Fox Foundation. We're trying to adapt the internet for clinical research studies using web-based data collection tools. This will allow us to dramatically lower costs and increase participation of people who might not be able to come into a clinical center who are interested in participating in a trial. I think I'll skip through this, but this study is recruiting currently. We're particularly hoping to be able to recruit additional control subjects. We're comparing, in a nutshell, this study looks to compare standard methods for collecting risk factor information, so that would be by interview or by paper, with risk factor information collected on the internet. So it requires people to take lifestyle history questionnaires several times. And this is just quickly, this is what the website looks like. An individual who signs up for the study would log into 23andMe's website and conduct some of our surveys online. These are surveys about head injury, pesticide use, use of caffeine, smoking, things like that. This is an example of one of our pesticide questionnaires. Another very important project that we're doing here is trying to standardize exposure history information, exposure history questionnaires around the world. And we are one of the foremost centers doing this. So investigators around the world are looking to us for leadership in this, and we'll begin using our instruments so that we can collect information in a very standardized way. So some of our newest work that we're just starting up is to look at biomarkers for Parkinson's disease. Now, a biomarker is a biological indicator. It could be something that we measure, say, tapping speed, heart rate, a compound in the blood, other bodily tissues. It can be anything, but it's something that is related to the disease process or response to treatment. And olfaction is a very interesting biomarker that we're including in many of our studies. The reason we're doing this is that it's been shown in a number of studies, and in particular in a study called the Honolulu Aging Study that has followed thousands of elderly men in Hawaii for about 40 years now. They found that impaired olfaction, which is very simply tested with a kind of a scratch and sniff test, it can precede Parkinson's disease by at least 4 years. So we're hoping that we can use olfactory testing as one of our biomarkers to predict the onset of disease before it presents, and also to track progression. Another biomarker that we're recently discovering, really, and are hoping to get funding to investigate further, is to look at heart rate variability. Now, heart rate variability, I'll talk about in a second, but it's a really fabulous way to study Parkinson's disease. We think that we can detect differences in people's heart rate just with a standard, simple EKG like you might get at an annual physical exam, that we can detect Parkinson's disease years before it presents, just through a simple analysis of an EKG. Another biomarker for Parkinson's disease that may be related to changes in heart rate variability is sleep disorders. Now, sleep disorders are very common in Parkinson's disease, but some sleep disorders may precede Parkinson's disease by several decades. This is an example of HRV's heart rate variability assessment using a simple EKG strip. And it's representative of changes to the neuronal innervation of the heart. And it's our hope, and we're currently submitting several grants to study this further, it's our hope that we can use this tool to identify people at risk for Parkinson's disease before they develop it. And we compared heart rate variability in a pilot study here, and this is about to be published in a journal called Movement Disorders, and we discovered that in people with a certain sleep disorder called REM behavior disorder, who did not have any signs of Parkinson's disease, that we were able to identify changes in heart rate variability that may predict that they will be getting Parkinson's disease within the next 5 to 10 years. Now, biomarkers allow us to diagnose disease before it occurs, and this is really important because we believe that therapies to prevent Parkinson's disease are only going to be most effective if we implement them as early as possible. So early diagnosis is really important. And we can also use biomarkers as endpoints for clinical trials to measure medication effectiveness, and this encourages pharmaceutical companies to put more effort into developing treatments for Parkinson's. So I'm going to finish up here talking about one study that is just getting underway, and there are some other people in the back of the room. Linda Reese and Dr. Liang is going to be talking about this study as well. It's called PPMI, the Parkinson's Progression Markers Initiative. This is a study that's getting underway with funding from the Fox Foundation. It's a worldwide study. There are 14 sites in the US, 4 in Europe. And all sites are going to collect data in a similar manner. Some of you may have heard of some recent reports about a breakthrough in Alzheimer's diagnosis using collection of bodily fluids and a systematic neuroimaging, so imaging of the brain, to predict Parkinson's disease. And we're trying to do the same thing in Parkinson's. So this is just getting underway. It's a big undertaking, a really big undertaking, even for Fox. And I don't have time to go into this in any detail, but I really hope that those of you who are interested will take some time to talk about the PPMI study with some of the folks in the back of the room, or with me, or Dr. Liang during the break, or after this session is over. How can you help? We really need control subjects for our research studies. Patients, people with Parkinson's disease are really motivated, and are fantastic about participating in research studies. But what people don't recognize is that for most of our studies, we also really need to recruit individuals who don't have Parkinson's disease to serve as a comparison population. And we need this for many of our studies. We're actually, if anyone's interested in considering participating as a control subject in our study, we've got folks in the back of the room ready to recruit and enroll you into any number of studies this very day or in the future if you want to pick up some information. I want to thank everyone for coming. I'm sorry I had to go so fast, and if you have any questions about any of the work we're doing or have done, please flag me down. Thank you.
Thank you very much. I'd now like to introduce Dr. Grace Liang. She's the head of our clinical trials program here.
Thank you, Jonathan. Can you hear me in the back? Okay, good. So as Jonathan mentioned, I'm one of the movement disorders neurologists here in our clinic, and I see many familiar faces. So I'm also heavily involved in our clinical trials program, so I'll just give a brief overview of the history of our program here at the Institute, as well as some ongoing projects that we have. So really, clinical trials have been an integral part of our whole mission and purpose as an institute from its beginning. We have been heavily involved in the academic realm, as you know, in terms of research for Parkinson's, and a lot of this has to do with how we can bring these research results to patients directly. And that's where clinical trials come in. So really, clinical trials are the way that we can bring discoveries from the lab to the patient. Because while there can be very many exciting findings in, say, a cell culture or in an animal model of Parkinson's disease, it's really not exactly the real thing until you see if it works in somebody who has Parkinson's disease. So the only way to really find that out is to have people who have the actual disease involved in looking at these different treatments and different aspects of the disease. So on the one hand, the research findings and discoveries can come to the patient and be, for example, approved as a new treatment or a new diagnostic test, ultimately through clinical trials. But on the other hand, in itself, it also brings a lot of valuable information about the disease, because through the clinical trials, we take so many careful assessments and measurements, or collect a lot of data and information about what happens over a long period of time for the patients. And certainly, we know that each patient can be very different, but when we look at a large group of patients going through certain procedures or certain tests over time, we can start to figure out these kinds of patterns that emerge. And a lot of those insights can then be brought back to the lab and developed further into better and better treatments. In general, I would probably divide our clinical trials into three broad categories, and you've heard a little bit about one of them from Dr. Goldman's talk. So the first category relates to biomarkers or early detection of the disease, and that's a very important area that's being developed currently. Another area that is also very exciting right now is the area of neuroprotection, or ways in which we can try to slow down or to modify a disease progression over time. So we know that Parkinson's disease is a very gradual, chronically progressive disease and condition, function, but often it takes many, many years to reach any degree of impairment or disability. If we can find a way to slow that down even further, many people would not mind having just mild symptoms of Parkinson's, and if it could stay stable that way. So certainly this area is a very critical need in our field right now. And finally, the last category is symptomatic therapy. So once you have the symptoms of Parkinson's or have the diagnosis of Parkinson's, what can we do to really improve or relieve those symptoms and help with day-to-day function and overall degree of independence? In terms of our studies looking at various biomarkers or trying to detect ways of identifying Parkinson's early on, we have a number of different studies that we have ongoing or have done fairly recently. And many of these are parts of collaborative multi-center national or international efforts that we participate in. The first two are looking at, for example, imaging and other biomarkers that can help to detect change in Parkinson's or predict Parkinson's disease early on. So, for example, the POSTEP study was a study of people who had very early Parkinson's disease, and they are being followed over many years with repeated scans. So this top picture here shows what's called a Spect Scan, and it looks at the uptake of dopamine in the brain. And in people with Parkinson's, the uptake of dopamine is significantly reduced. So if we can follow the change over time, it would give us a better measure of how much dopamine is in the brain or how functional those brain cells are. And the PARS study is a similar study, but looking at a different population. So in this study, we actually recruited a number of family members of Parkinson's patients who they themselves do not have Parkinson's disease. But as Dr. Goldman mentioned, there may be a slightly increased risk of Parkinson's in family members because of some genetic factors. So we're looking at a number of many different factors such as smell function, sleep patterns, mood questionnaires, or other kinds of things, as well as these scans and neurological exams over time to see if we can predict if there are some abnormalities in some of those areas, will that person go on to develop Parkinson's disease down the line? So that's a very important study for us to be able to pick up earlier, if possible, or predict who might be at risk for Parkinson's down the line. There are also a number of other studies, such as the PPMI study and some of these other studies that look at, say, biological fluids or tissues. And in the same way, if we can measure with a blood test or, for example, sampling some cerebrospinal fluid, there would be some chemical markers, perhaps, in our own systems that we can track or make a better accurate diagnosis. So a lot of my patients ask me, why is it that you can't tell me for sure if I have Parkinson's or not? Or isn't there some sort of test or MRI scan or something that will tell me definitively if I have Parkinson's? Well, the brain is a very well-protected organ in the body, and it's not so easy to have access to that. So if we can, through a simple test like getting some blood or urine, or in the PPMI study, we will be looking at cerebrospinal fluid through a lumbar puncture. That way we can get a little bit closer to what's actually happening in the body and hopefully be able to track over time how the brain cells are actually functioning. So one analogy I use is that the measures that we have for looking at progression right now are very, very blunt instrument. So it's almost as if you're trying to see how quickly somebody's walking on the ground from, the height of an airplane. If you're flying in an airplane from 35,000 feet and you're trying to identify how quickly somebody's walking from below, there's no way you can really tell the speed because, your resolution isn't good enough. You can't see that far. So that's almost what we're dealing with right now. What we can see are the outward signs of Parkinson's. We can sort of measure clinically how fast you walk or how fast you move or how strong your tremor is. But to really see what's happening inside the brain and the molecules in the brain is much more difficult. So we are hoping that through these studies we can really get at that question. Another way of attacking this too is using some other objective measures of change. So there's this little device here that's been under development, funded by the Kinetics Foundation, looking at things like measuring tapping speed or speech qualities and so on. So something that perhaps I might not be able to detect, but with a machine you might be able to detect subtle changes over time much more sensitively. So those kinds of things are very important to our development of clinical trials and research endpoints. In the area of neuroprotection, we have quite a number of trials ongoing, and some of these are currently enrolling. So the top two are called the Qe3 trial. It's short for coenzyme Q10, looking in very early Parkinson's patients. So people who have not been treated for their symptoms of Parkinson's, who are fairly mild at this time, but we are looking to see if high doses of coenzyme Q10 may help to slow down the change over time. So there was a very small study that was done many years ago that suggested there might be some benefit, and there's also some laboratory data that suggests that the coenzyme Q10 may be beneficial for the energy centers in our cells. Cartoon of the mitochondria, which is within each of our cells, and we know that Parkinson's disease affects the function of the mitochondria. Creatine is another component, another compound that's being studied in this NET-PD study. Both of these are NIH funded, so they're nationally government funded, very large trials ongoing, looking over a number of years to see rate of change. And again, creatine is important in energy production within the mitochondria as well. The other two studies, one is called isradipine, STEADY PD. Isradipine is a calcium channel blocker that affects signaling pathways within the brain cells. And there's been some quite exciting data from Northwestern looking again at an animal model of Parkinson's where it looked like use of isradipine was protective of the dopamine cells and may even help to reverse or revert them back to a more youthful or a juvenile state. So if that's a way that we can use to help compensate for the changes from Parkinson's, that would be very useful to know. Rasagiline is currently approved for the use of Parkinson's disease treatment, but we're also testing it in people who have multiple systems atrophy, which is not exactly the same as Parkinson's, but is a similar neurodegenerative disorder. So again, these are very exciting studies that we have ongoing. Certainly, we know that symptomatic treatment is very important for overall function within Parkinson's, as well as degree of independence and things like that. So we classically define Parkinson's as having some typical motor features. So the cardinal features of Parkinson's are a slowness of movement or bradykinesia, tremor, usually at rest, some rigidity or increased muscle tone, and also gradually balance difficulties or walking difficulties. So those are the typical features of Parkinson's, and over time, with treatment, sometimes you can have some fluctuations where you have periods where the medication works well and other periods where the medication doesn't work so well, or dyskinesias, which are some excessive or involuntary movements that can be stimulated by medication treatment. So these are well-known features of Parkinson's disease. Also, we're increasingly aware that there are many non-motor features of Parkinson's. So, for example, we know that sense of smell is something that's very early on. Even before the motor symptoms come on, people can lose their sense of smell quite dramatically. And there may be other sensory features, for example, different types of pain or muscle cramps. There are a lot of other areas like sleep disturbances or fatigue can be a common symptom in Parkinson's. Some of these things may not be completely specific to Parkinson's, but are much more common in Parkinson's than in other people who are of a similar age. There may be other things like autonomic dysfunction. So, for example, blood pressure fluctuations, problems with GI function like constipation, urinary symptoms, all of these areas may also be contributed to by the Parkinson's. And finally, there may be some psychological or cognitive changes that go along with Parkinson's, such as mood changes, anxiety or depression, or sometimes difficulty with memory or thinking processes. So all of these factors certainly play a very important role in a person's quality of life as well. For treating the motor symptoms, we have certainly very good treatments that are already available and are approved. So it's quite fortunate that, over the past 10 or 20 years, we have had a number of many new drugs come into our armamentarium that we can use to treat Parkinson's. But there are still, certainly many needs and certainly new drugs under development. So some of the trials that we have ongoing right now are the Untitled Trial is looking at the use of rasagiline as an add-on treatment to people who are already taking a dopamine agonist like Requip or Mirapex alone. Rasagiline is approved for use as a monotherapy by itself or in addition to levodopa, but it hasn't been tested completely when added on to a dopamine agonist. So this is for people who aren't sufficiently treated with just the one drug and haven't had any exposure to rasagiline or selegiline in the past. There are also two other studies called Motion and Settle looking at a new drug that's not yet approved called safinamide. And that's a drug that's in the same class as rasagiline, but it may have some other different mechanisms of action. So we're looking to see whether it, in addition to a dopamine agonist or to levodopa, may also be beneficial to help control the symptoms better. There are also some newer levodopa formulations that have come under development and are being tested. So we know that Sinemet or carbidopa levodopa is sort of the gold standard treatment for Parkinson's for a long time. And it's been tried and true. It's certainly one of the most effective treatments for Parkinson's right now. But the problem is that over time, the duration of its effect gets shorter and shorter or may become more unpredictable. So, for example, people may have periods where it works, and then as the level wears down, then the symptoms come back. So some companies have developed ways for our body to handle levodopa or metabolize it in a way that it could potentially last a little bit longer and help to smooth out some of these fluctuations or ups and downs in the treatment effect. So Impax and Xenoport are two of these newer formulations that are being tested now for that purpose. Finally, for treating dyskinesias, we know that this can be quite disabling. Sometimes people, even though the medication is working, they may have excessive movement or involuntary movement related to the medication effect. And sometimes that can hinder people from increasing their medication to better effect because of the dyskinesias involved. So there's a new drug developed by Novartis that is also being tested to help control the dyskinesias. So these are also ongoing and enrolling trials, and we will have information or somebody to answer these questions, or you can come talk to me if these are interesting at all. I mentioned too the non-motor symptoms, for example, loss of smell. Now, a lot of people may not think that loss of smell is a big deal. Certainly it doesn't cause a great deal of disability, but certainly it can affect your quality of life quite a lot. Your sense of enjoyment of food or being able to detect your environment around you can be impacted quite a lot by sense of smell. And another thing that we are interested in that aspect is because it seems to be a very early marker of Parkinson's disease changes. So if we can detect some of these changes or improve that aspect of Parkinson's, it might give us an indication of something that's actually going on in the brain as well. So we have a study going on now looking at the use of rasagiline to see if use of that might help to improve sense of smell. And we're looking for people who have not been taking rasagiline or selegiline at least for the past year. Hallucinations can sometimes be associated with Parkinson's, especially in association with cognitive decline or memory problems. So there are some new drugs that are being looked at to treat that aspect, because that in itself can be obviously very upsetting for the patient and for family members or caregivers who have to be involved in their care. Many of the anti-hallucination drugs out there aren't safe for Parkinson's patients. They can worsen the Parkinson's disease. So if we can find a safe treatment for it for our patients, that can help quite a bit to improve quality of life again. And the cognitive aspects are certainly very important for us to monitor and to intervene on if that's an important part of the patient's overall well-being. I covered a lot of ground, I know, in a short time, but what are things, how does this really impact you? Well, certainly just being here is a very important part of it. So finding out about the current research that's going on, staying informed and educated is a very important part of what helps the whole process. So you've seen certainly that Michael J. Fox has taken on this very important role of being being such a great advocate for Parkinson's, just putting himself out there and his own experience. And each of you can also do the same thing. So learning about the research that we do here, sharing it with other people in your family or in support groups or in your community is important overall, just to increase awareness of Parkinson's and all the major hurdles and challenges and progress that we're making in the field. I think that's very important. So you're staying informed and educated, and helping to inform and educate others is such a critical part of helping us in our work. And of course, we're always looking for volunteers. So some people who might be a candidate for these trials, or even non-patients. So again, healthy controls are certainly people we could use to help us further the research more quickly. One of the big hurdles, and we always hear, and we feel the same way, that the research is so slow. Why is it that we hear about something and then for years and years nothing comes of it? Well, a big hurdle that we know is that we just don't have enough people volunteering for these kinds of studies. We would do a lot better and be able to get those results or bring the treatments to you much faster if we were able to fill the studies for recruitment much more quickly, get all the people that we need involved, and then be able to find the results as quickly as possible. And so that speeds up the whole process overall. So if you're interested, I'll be available. Certainly our staff and our nurses, I saw Dr. Brandenburg here, so you can certainly talk to us or give us a call. We also have a web page or an email address up here, trials@thepi.org. If you want to send us an email or give us your information, we'd be happy to take that down and give you more details about the particular studies. So thank you very much for your attention, and I'll be happy to talk to you individually or answer questions down the line. So thanks a lot.
So at this time, I'd like to introduce the new kid on the block. He's our new scientist here at the Parkinson's Institute. His name is Jeremy Nichols.
All right, so I'm a biochemist and molecular biologist, and I'll tell you a little bit about how I think about disease and how we're trying to work towards a therapeutic and a cure. All right, so as a biochemist, I came to the Parkinson's disease field anew, and this is how I view disease. So we have two different ways Parkinson's disease arises. One is through sporadic or idiopathic means, and one is through familial. But for the purposes here, I would like for us to think that the symptomatic manifestations of this disease is similar between the two. And one of the big questions in the field is, what are the pathways or the highways of cellular signal transduction that are disrupted in disease, lead to the observed pathological hallmarks of PD, which is the dopamine neuron loss, and which leads to disease presentation? I like to think that both there's a common pathway that is shared between sporadic PD and familial PD. And as Sam told you, that we think there are environmental factors, and now there's also inherited mutations, and that's the same thing as familial disease. And it is likely that these pathways that are disrupted in familial PD mirror those in sporadic PD, and therefore understanding one will shed light on the other. And I like to think of it as if we all have a house on stilts. If you lose one of those stilts, it might just be that the house is not as stable as it used to be. And what we want to do is find a way to put those stilts back. Over the years, there's been an association of many genetic loci with Parkinson's disease, and here are 16 as well as many others. And this is through large-scale genome-wide sequencing efforts that have taken place all across the world. And I just want to point out a few of them here to you. One of them is alpha-synuclein, and it has been found to be 2 different forms, and Sam told you about 3. And alpha-synuclein is going to be important especially in idiopathic disease, and following me will be Ian Irwin, and he'll tell you a little bit about the work that he's doing to understand how we can treat disease based on alpha-synuclein. And also there are 2 other genes. There's PINK1 and LRRK2. These are enzymes, and enzymes are good because they catalyze reactions, so they do something in the cell. And if you're doing something and you're doing it wrong, we think that we might be able to develop a drug that might tell you to stop. And we think that might be a way to a therapeutic. And so if I'm going to tell you about the biochemistry of LRRK2, just to tell you what biochemistry is, it's actually the conjunction of biological chemistry. So if you think about chemistry, there's chemists in the lab that use tiny molecules, and they're cooking them in their beakers. But what I'm concerned with are the proteins, the products of the genes that are encoded by your DNA. I want to know the details of the processes that happen in the cell and what is going wrong in Parkinson's disease. And so what is LRRK2? So LRRK2 is actually an acronym for leucine-rich repeat kinase 2. So leucine is a building block of a protein, and LRRK2 just happens to be having a preponderance of these building blocks. And this is an enzyme, as I said. It's a kinase. So a kinase is an enzyme. In science, we call enzymes ACEs. And so if you just take kinetic and ACE and put it together, it's a kinase. And kinases catalyze a reaction that the details aren't important, but what it does is transfers this molecule to a protein in a cell. And it turns out that the majority of the cellular processes are regulated in this manner. And to underscore the importance of this, in 1992, Edwin Krebs and Erwin Fischer received the Nobel Prize for the discovery of reversible protein phosphorylation. And protein phosphorylation is mediated by kinases. And in 2009, the Lasker Award was given to Nick Leiden for the development of Gleevec, a selective BCR-ABL inhibitor for chronic myeloid leukemia. And this is one of the first times that we were able to use a kinase to treat a disease. And it was a very successful development of a drug. And now this makes LRRK2, since it is a kinase, a very highly targetable molecule for therapeutics. So LRRK2 is found in about 1% of idiopathic PD and about 5% of familial PD. And in some isolated populations, we can have amounts of mutation rising to 40%. Most commonly found mutation in LRRK2 increases the activity 1.5 to 2-fold. And disease presentation in most all cases mirrors that seen with idiopathic PD. And so I want to know, is disrupted LRRK2 function similar in idiopathic PD as well as familial PD? But there's a problem in that. And I'll tell you about that in a minute. So if we want to take what LRRK2 looks like and stretch it out in a linear fashion, it's a very large molecule. It has many domains, and that's what these boxes are. But what this is used to represent is that there are many, many mutations that have been found to be associated with Parkinson's disease. The ones that are boxed in red are found to be pathogenic, so if you inherit this disease, it is highly likely that you're going to contract Parkinson's disease. The most commonly found mutation is this G2019S mutation. And this is the one that causes the increase in kinase activity. Now, I won't go through how we do this, but if you just take these bars as a representation of how much activity LRRK2 has, we've assayed all these different mutations. And this is the most commonly found mutation, and this is what the wild-type enzyme gives you. And so just the increase in bar size tells you that there's an increase in activity. And we've also identified another mutation that's associated with Parkinson's disease that also increases activity. And what we'd like to do is to facilitate the development of a drug that might be able to modulate this activity to bring it down to wild-type levels, and maybe that can serve as a therapeutic. LRRK2 has been studied for about 6 years now. There are over 500 publications, but one of the problems is that only about 20% of those are reviews, so it's just scientists telling other scientists what they know about it. And so we're all just talking to each other about the same thing because there's a lot not known about the enzyme. And so this is a typical signal transduction pathway inside the cell. All the little blobs don't need to be explained, but I just want to let you know that to develop this scheme, each one of these is a protein. Each one of these circles is a protein, but to develop this took years and years and years to describe. And all of these arrows point to kinases. And so there's multiple entry points for potential therapeutics. And what we want to do is try to put LRRK2 in a signal transduction pathway. And if we can define this, we might not only be able to develop inhibitors to LRRK2, but there might be other pathways that LRRK2 intersects with that we could also target for development of therapeutics. We're trying to find out what in the cell, what outside the cell might talk to LRRK2, tell it what to do, what inside the cell might tell it what to do, and then what is LRRK2 talking to? Is it talking to itself, or is it telling something else what to do? And it's these processes that may be disrupted in Parkinson's disease. And the problem is that we have question marks everywhere we turn. But what we'd like to do is develop a signal transduction pathway and describe it so that we can work with big pharma to develop therapeutics. And we're currently interacting with a lot of drug companies to work on LRRK2 inhibitors and also develop these signal transduction pathways. So we're trying to bridge the gap between academia and industry so we can move forward with therapeutic. LRRK2, a disrupted LRRK2 does something wrong, and then this leads to disease. In idiopathic disease, we know that something goes wrong, and then you get disease. But what I think is that there's an intersection between the two, and if we find out what LRRK2 is doing and focus on developing therapeutics for this enzyme, it might be a way to a therapeutic. And so the way that my lab is working towards getting there is doing biochemistry, right? So we're trying to find out exactly what the molecule does. and then developing cell models and animal models so we can test drugs before we start moving into clinical trials. And Birgit in a little bit will tell you about some of the ways that we're going to try to merge her techniques and my techniques so we can have a really good handle on how these drugs might affect Parkinson's models so that we can move them into trials in the clinic so we can reach a therapeutic and potentially a cure. So that's how a biochemist thinks about disease, and that's how we think about defining the stepwise processes that we might attack so we can get to a cure. So that's all. And if you have any questions, I'll be around, and I'll be at the end of the question and answer session. All right.
Next on our agenda is Ian Irwin, I think. Is that right? Ian? Oh, there you are. I'm sorry. Ian was actually one of the first employees at the Parkinson's Institute. Those many years ago, about 20 years ago, I think, or so. He left the Parkinson's Institute for a period of about 10 years or so, and he's rejoined us in the last couple of years, and he will explain his role in the Institute in our basic research facility. Ian Irwin.
As Jonathan mentioned, I was actually employee number 1 of the Institute in 1984. And I've worked in the pharmaceutical industry. I worked here for about 15 years and then worked in industry for a while, mostly developing drugs. So working in a preclinical and early preclinical drug development stage. And I rejoined the Institute about a year ago. And I'm working, again, on trying to demonstrate animal models and models of disease that we can test pharmaceuticals. We work very closely with industry in what are called preclinical studies. These are studies before we get to man. What I'm going to talk about today is a program that was originated here, and I'm going to focus primarily on this program of alpha-synuclein. Again, you've heard from Sam talking about clinical research, Grace talking about the clinic and clinical trials, and now this afternoon we're talking about basic and preclinical research. And the Institute is really a unique structure. It really has these 3 arms, and I depict this as a way discoveries flow back and forth and information flows back and forth. Clinical research and observations made during clinical trials and other general medicine really reflect back, develop a lot of ideas about testing drugs and new ideas about targets for drugs. And the basic research department does work with preclinical and basic work. So that goes everywhere from very fundamental studies like Jeremy's talking about, really talking about very new areas and trying to understand the LRRK2 pathways and how we might exploit those for treatment of disease, to doing preclinical studies that just demonstrate proof of concept before we go into the clinic. So I'm going to talk today about the Alpha-Synuclein Program, our Alpha-Synuclein Program. Why alpha-synuclein? Why is that a target? Well, Jeremy did a very nice description of overview of the genetic causes of Parkinson's disease and familial PD, and Brigitte will talk about this, I'm sure, again. But the vast majority of the 1.5 million PD patients in the U.S. have an idiopathic form of the disease or a sporadic form of the disease that is not inherited. So when you look for what is common amongst the PD sufferers, it's been known for some time, actually discovered by Louis. Louis finished up his PhD about 100 years ago, 1910. I was just reading about him today. He also was at the same institute with Alois Alzheimer, and both Creutzfeldt and Jakob were there together. So these are very big names in neuropathology. And what Louis did was he was a neuropathologist and neurologist, and he studied Parkinson's brain using a microscope. And what he noticed that were these eosinophilic inclusion bodies, these bodies within a neuronal cell with a dense core and a halo. And they really are the primary marker for Parkinson's disease. And they appear throughout the peripheral and central nervous system. I think what we struggled with since in 1982 when I first became involved in PD research. We're trying to understand the commonality where all the Lewy bodies occur, because they occur throughout the central and peripheral nervous system, even in the gut, in other areas of the brain, locus coeruleus. And it's not just areas that contain dopamine. It's areas that are non-dopaminergic, that are cholinergic, and adrenergic as well, too. So it was a mystery, really. And with the first cases of familial Parkinson's disease, it was found that those were due, the first families, was due to a mutation in the alpha-synuclein gene. Well, knowing that, people made specific techniques to now look for alpha-synuclein in the brain. And all of the areas showed this aggregated conglomerate of alpha-synuclein in Lewy bodies, and not just in Lewy bodies, but in neurites also, in peripheral neurites. And what we've learned about the Lewy body, it contains about 40% alpha-synuclein, primarily in the halo. This is an EM of a Lewy body, and this is an even higher magnification showing these areas, this area of the halo. And the halo consists of these alpha-synuclein fibrils. And what we've come to learn is that alpha-synuclein exists, the actual function of alpha-synuclein is not fully known. It's thought to be involved in neurotransmission and vesicular release in neuron cells, but it exists in a soluble and a folded conformation. And that alpha-synuclein, and we can see this in the test tube, will actually unfold and then refold into aggregates and fibrils. So these dense aggregates of multiple units of alpha-synuclein. And in fact, the genetic mutations that lead to Parkinson's disease all produce an alpha-synuclein that, if you put it in the test tube, is much more prone to aggregation. There's another genetic source of PD, which is duplication and triplication. So more than the normal number of genes, additional genes of alpha-synuclein. So it's simply an overproduction of the normal alpha-synuclein. We know that the more alpha-synuclein that's present in a test tube, the more likely it is to form aggregates. And so we've exploited this ability of alpha-synuclein to form aggregates in the test tube. You simply can put alpha-synuclein, recombinant human alpha-synuclein, in a tube and incubate it at 37 degrees over time. And this is hours. And you can do this in a multi-well plate. And what happens is it goes from this soluble form to an aggregated form. And we have a tool, a chemical that reacts with the aggregated form to fluoresce, to glow. And we can measure that fluorescence. So this is the basis of an assay to measure the effect of compounds on alpha-synuclein aggregation. And so what we did was you can buy libraries, these wells, multi-well plates, libraries of known chemicals, small molecules. And you can incubate alpha-synuclein in the system and then add the test compound. And what we did is we did this with over 3,500 compounds. And you can see here, this is alpha-synuclein alone. And this is alpha-synuclein in the presence of a test compound. It completely blocked the aggregation, and when you look at the formation of aggregates, there are none. Not only that, but you can do this experiment another way. Instead of adding the compound at the beginning and looking for the formation of fibrils, you can develop the fibrils, and now you can add the compound here. And that's what we've done in this experiment. So we've allowed the aggregation to occur, full fluorescence, then add the test compounds, and this shows some 6 compounds that actually dissolve or disperse the aggregates. So based on this tool, we've developed a plan, a project plan, and a certain preclinical steps. What we're really out is to find a drug that can slow, stop, or even reverse the aggregation of alpha-synuclein in Parkinson's patients, and thereby either slow or even reverse the disease. And this is the current status. Now, we've been working on this for a number of years. This is not a new project, and we've encountered some challenges, but we are moving forward on it. We've completed the screening of the large libraries. We've got 80 compounds, including some 6 to 11 compounds that actually dissolve. So this is a very high concentration. The next step is to compare the compounds at different concentrations to identify the most potent compounds. Why is this important? Well, you wouldn't want a compound that would require, should take tablespoons and tablespoons. It would be not a practical drug. So we're really looking for the most potent compounds. So we'll identify the best compounds. Out of that, we're looking to come up with 20 or so compounds. And then we just submitted a grant to Michael J. Fox to work with Jeremy to look at these compounds in cell culture. Jeremy's developing a cell culture model of the alpha-synuclein aggregation so we can see whether we can protect living cells against the effects of alpha-synuclein overexpression. And finally, we need to determine whether the compounds are really drug-like or not. For example, I may talk a little bit about this, 98% of small molecules don't get into the brain, so a compound might appear to be an ideal agent, but it would be inadequate. So these studies define oral bioavailability, that you can swallow the drug and it goes into the system, that it lasts long enough that it will make a drug and that penetrates the brain. And ADME stands for absorption, distribution, metabolism, and excretion. This is basically the PK of the compound. And finally, we need to test the drug in an animal model of alpha-synucleinopathy or alpha-synuclein overexpression. I was talking about potency. Well, here's a comparison of 3 compounds. So what we do is we put increasing concentrations of the compound in with the alpha-synuclein system. So this is no compound, maximum fluorescence, maximum fluorescence, maximum fluorescence. This is alpha-synuclein aggregating. Now you can see for this compound, TPI-6, at 1 micromolar, we're more than 50% inhibiting the process. This is a very good compound. At 50 micromolar, completely inhibited, and at 25 micromolar, also completely inhibited. Here's a compound that I say is intermediate potency. You can see it's not quite as potent as this one, so it's only about 75% inhibited at 1 micromolar, and it doesn't fully suppress alpha-synuclein aggregation, even at higher concentrations. And here's a low potency compound. Even if this is 25 and 50 here, it does not suppress fully alpha-synuclein aggregation. So it's very important for us to put all our leads through this process to distinguish which are the most potent ones. As I said, more than 90% of small molecules, when you take them, they just don't get into the brain. It's a major reason for failure of CNS drugs. So we need to make sure we can find compounds that do get into the brain and act in the brain. Finally, we have developed an animal model to test our best leads. We can't do this with very many compounds. 3 to 5 compounds is what we're estimating. But you can see here, what we've done is we've injected essentially the gene for alpha-synuclein into the rat brain. This is a control. This is the gene injected. And this is the substantia nigra, tyrosine hydroxylase staining in the substantia nigra. You can see that on the injected side that received alpha-synuclein in both these animals, it severely damaged the dopaminergic cells in the nigra. And here is the loss of dopamine and the metabolites of dopamine, DOPAC and HVA. It produces a 50% loss of striodopamine. So this is the overall sort of schema for drug development. Phase 1 is when a drug goes into normal humans, the early stages of testing. And then phase 2 is limited testing in patients with the disease. And 3, we're right here right now. We're trying to get to animal testing. But I think this is a very exciting project. I mean, it's probably, right now, I think that LRRK2 is a very important target. But right now, I think that alpha-synuclein aggregation is probably the primary target for new therapeutics. Thank you.
So our final presenter will talk about one of the most exciting things we're doing now, and I'll let you tell you about it. This is Dr. Birgit Schüle.
So I called it Future in Stem Cell Research. I hope there's a great future for stem cell research, and I specifically talk about induced pluripotent stem cells from patients for understanding Parkinson's disease. I want to start with 2 introductory slides. One is on definition of stem cells, and the other one is talking about differentiation itself. So what is the definition? Stem cells are the body's master cells. They have the ability to grow into one of the over 200 different tissue types of the human body. And these cells are unspecialized, or they're also called undifferentiated. And they retain the ability to divide throughout life, and they self-renew. And then they do contribute to the body's ability to renew and repair tissues. And that's something that we want to use in the future to cure or treat human diseases. So this is another thing that's very important for this talk. It's a dogma, I call it the old dogma on cell differentiation. Till 3-4 years ago, everyone thought it's only a one-way street. So you start with a stem cell, and the stem cell differentiates into a specific tissue type. Stem cell into a liver cell, stem cell into a heart cell. But we were thinking there's no way back to revert that back into a stem cell. I mean, there are always exceptions, but just the ballpark picture. You start with a stem cell, you differentiate, they have their function, and you're done. You can't go back. But we know now that we can revert to an undifferentiated state, or we can rejuvenate those differentiated cells into stem cells again. And this was kind of talked about in 2008, and it was really one of the scientific breakthroughs. And the Science Journal really gave this new technology one of the highest honors. And so we can now produce made-to-order cell lines by reprogramming those cells, and specifically from patients with certain diseases. And I mean, we hope that these cell lines offer in the future tools for understanding the disease better, but also we hope that someday we can cure some of these difficult to treat diseases like Parkinson's disease. So this is how we think about it. We start up there with a patient with a specific disease or an injury or disorder. We take skin cells, we take a skin biopsy and culture these cells in a culture dish. We just have to add a few growth factors and media, some salts and sugar. So my little son is calling it soda. It looks like soda. It's kind of this reddish orange solution that you have to feed the cells with. So we're giving those cells the soda, and then we bank those cells, first of all, the skin cells. The next step is called the epigenetic reprogramming, the yellow box. And that's where the interesting thing happens. That's where you revert the differentiated skin cells back into stem cells. Once we are there, you see at the bottom, it's called pluripotent stem cells. At that point, you're free to use those cells to differentiate them into the tissue types of interest for your research. And we are interested in the dopamine-producing nerve cells. So in the culture dish, we differentiate those cells back into neurons. And then there are really two ways to do further research. One is the cell replacement or autologous transfer of those cells back into a preclinical model, or hopefully at some point back into a human brain. And then the other area is in vitro research. So understanding disease mechanisms, using these cells to do drug screening. So those are two big areas we are interested here. So we are not primarily interested in cell replacement. First thing we have to understand the disease a little better, maybe find drugs before we can really say we take those cells, put them back into a human brain. So this is how we do it. We start with the skin biopsies, and these skin cells are also called fibroblasts. So you can see I'm taking skin biopsies, and then in the culture dish, the cells look like this. So at the bottom, you see written skin biopsy, this darker area. This is a piece of tissue. And then there are two different cell types growing out. And the first cell type is called keratinocyte, and the second that we then use for reprogramming or rejuvenating the cells are the fibroblasts. We bank those, and maybe of note here is we don't use human embryonic stem cells here at the Institute. This is all from patients that we see in the clinic. So we don't use any human embryonic stem cells, which is a big kind of ethical discussion. So we don't have that problem here. So this is a scheme how we then induce them and reprogram them. So at the left, we start with the fibroblasts again, or the skin cells. Then in this case, we are adding viral vectors that produce certain proteins. And you've heard about proteins before, and I know you're a very smart group of people here. You know what that is. So we only need four different proteins, and those are enough then to convert those skin cells into stem cells again. And then at the end, when the cells are happy and they are fed every day, then we see at the right bottom the stem cells. So once we have those stem cells, we want to differentiate them into neurons. And we start there at the left in the middle with the stem cell colony. And then we have a specific assay that allows us to differentiate those cells into dopamine-producing nerve cells. So there are different steps. One is called embryonic body formation, so you have little balls in culture with probably a couple of thousand cells. Then you plate them down, and then they form these rose-like structures. You isolate those rose-like structures or neuronal rosettes, then expand them as neuronal stem cells, and then you add another cocktail of different growth factors into it. And at the end, after roughly 35 days, you have dopamine-producing nerve cells in the culture dish. It's a long process, I have to say. You don't do that overnight. So this is just an overview, and it shows all the different steps. So we start with a skin biopsy, grow those skin cells first, then bank them. Then we have the second step with the transfection of these reprogramming factors. We characterize our stem cells, and then we wind our way down to make dopaminergic cells. And at the end, we really want to use those cells for high throughput screening, looking at disease mechanism. And this is an interesting term that I just learned today. We like to do in vitro clinical trials. So in vitro means in a culture dish. So that's the hope that we can use these cells for. Okay, this is a thank you. And I really want to thank all the patients and volunteers that are participating in the study. And I've already seen a couple of people here. Thank you very much. I really appreciate that. And I also want to thank the lab, the people that are working on this program, Dr. Langston, who is the principal investigator. We also have very wonderful collaborators at Stanford University, the lab of Renee Ray-Opera and Theo Palmer. And then we have a corporate partner, Hyperion, and I want to thank these organizations for the funding and support. California Institute for Regenerative Medicine, Parkinson's Alliance, Bloom Foundation, and Danville Delegates. Thank you.